Welcome to Botany Talks. This class is about prokaryotic translation. In the last class, we discussed about the various components required for prokaryotic translation. Through this class, we can learn about the different steps involved in prokaryotic translation. What are the different steps in prokaryotic translation? There are three steps: initiation, elongation, and termination. These are the three steps in protein synthesis of prokaryotes. The first step is initiation. Initiation requires different components such as mRNA, formylated methionine-tRNA, 30S ribosomal subunit, 50S ribosomal subunit, initiation factors, and GTP molecule. First, look at the mRNA. mRNA is the information provider. Based on the information, that is codons, present in mRNA, a polypeptide is synthesized. There is a codon called start codon, and the start codon is AUG in all mRNAs. That is, the first amino acid to be added to a growing polypeptide chain is the methionine amino acid. In the case of prokaryotes, the methionine alone is not added. A formylated form of methionine is added at the beginning. But if AUG is present at the inner regions of the mRNA, in that time only methionine is added, but at the beginning formylated methionine amino acid is added to begin the synthesis of a protein molecule. Therefore, mRNA is the basic information provider. Then second one, what about Shine-Dalgarno sequence? This is a conserved sequence located upstream of the start codon in mRNA. This is a purine-rich region present in the mRNA molecule. At this region, the first initiation complex will bind, or ribosome will bind to initiate the process of protein synthesis. This is a specific feature of prokaryotic mRNA molecule. Upstream to every gene in prokaryotic mRNAs, we can see the presence of a Shine-Dalgarno sequence. Without this Shine-Dalgarno sequence, the translation process cannot begin. So, any mutation in Shine-Dalgarno sequence can stop the process of protein synthesis. Therefore, this Shine-Dalgarno sequence is a very important factor for prokaryotic translation. This is a purine-rich region. Mainly it consists of adenine and guanine adenine and guanine nucleotides. The second factor is formylated methionine-tRNA. This tRNA is present in cytoplasm. It will detect the start codon and get attached to the ribosomal binding site. Formylated methionine-tRNA is the only tRNA that will directly bind to the peptidyl site of the ribosome. All other new tRNAs will bind at the A site, while formylated methionine-tRNA will bind to the peptidyl site or P site of the ribosome molecule to begin or initiate the translation process in prokaryotes. There are two subunits of ribosomes which are involved in translation. Ribosomes are the organelles in which the translation process is carried out. 30S ribosomal subunit is the first subunit that attaches to the mRNA molecule. Following this, only the 50S ribosomal subunit will join to the 30S ribosomal subunit and forms the complete machinery for protein synthesis. The next factor is initiation factors. These initiation factors are necessary components for initiation of protein synthesis in prokaryotes. There are three different types of initiation factors in prokaryotes. They are IF1, IF2 and IF3. IF1 directly binds to the 30S ribosomal subunit. it will block the A site of ribosomes and helps the peptidyl site, and helps the formylated methionine-tRNA to directly attach to the P site of ribosomes. IF2 is very much required for binding of fMet-tRNA to the P site of ribosomes. Next, IF3. This is the component which binds to the 30S ribosomal subunit and attaches itself to the Shine-Dalgarno sequence of the mRNA and initiate the translation process. IF3 associated with 30S ribosomal subunit. IF2 associate with fMet-tRNA and IF1 associate with 30S ribosomal subunit. We can see this process one by one. Just this is the 30S ribosomal subunit. It is attached with initiation factor 3. This assembly will come and bind with Shine-Dalgarno sequence. This box represents the Shine-Dalgarno sequence. This is the ribosome binding site. RBS represents ribosome binding site. To this ribosome binding site, IF3 30S ribosomal subunit complex will come and attach. This Shine-Dalgarno sequence is a purine-rich sequence. It is having the sequence AGG AGGU. The second step is binding of fMet-tRNA to the start codon of mRNA. This is facilitated by IF2 GTP complex. Energy for this binding is provided by the hydrolysis of GTP molecule, and it is driven by IF2. Initiation factor 2 associates fMet-tRNA and helps it to bind to the start codon on mRNA. Next is the binding of 50S, that is the larger subunit of ribosome, to this complex. There is one condition. In an initiation complex, IF3 and 50S ribosomal subunit cannot occupy together. The release of IF3 signals the binding of 50S ribosomal subunit to this 30S ribosomal subunit. Only when the initiation factors and GDP are released from the 30S ribosomal mRNA complex, 50S subunit will come and bind. In this figure, you can see the assembly. This assembly represents the initiation complex for protein synthesis in prokaryotes. This initiation complex contains an mRNA molecule, this fMet-tRNA, 30S ribosomal subunit and 50S ribosomal subunit. All the initiation factors IF1, IF2 and IF3 along with GDP will be released from the initiation complex, pre-initiation complex, and final initiation complex is devoid of initiation factors and GDP molecule. The second process is elongation. In elongation, the actual synthesis starts and the chain will grow by adding amino acids together. In elongation, the first step is binding of a charged tRNA molecule to the A site of ribosomes. We already discussed that A site is first blocked by the initiation factor IF1. So, the first tRNA, that is fMet-tRNA, binds to the P site. When IF1 is released, now A site is free. So, it can accommodate a new tRNA molecule. Therefore, binding of a new Aminoacyl-tRNA to A site is the beginning of elongation phase of translation. Next is the formation of peptide bond. This peptide bond is formed by the activity of an enzyme that is called the peptidase transferase enzyme. Next is the translocation of ribosome. Translocation of ribosome means ribosome molecule will move towards the 3' end of the mRNA molecule. It will covers each codons. It will move 3 nucleotides forward, and it will move throughout the mRNA molecule until it reaches a stop codon in mRNA molecule. And only when it reaches a stop codon, translation process will be stopped. Until then, the elongation phase will continue. What are the different events that occur in an elongation phase? In this picture, you can see a 30S ribosomal subunit, a 50S ribosomal subunit containing 3 different binding sites for tRNA molecules. The first site is the A site. Here, a new tRNA, aminoacyl-tRNA molecule can bind. The second one is the peptidyl site. Here already a tRNA is present, that is fMet-tRNA is present at the beginning. Then next is the E site, this is exit site. First, tRNA selection. Based on the codon present in this mRNA molecule, the appropriate tRNA from the cytoplasm will bind to the codon or A site of the ribosome. This A site tRNA has an amino acid which represents the codon present in the mRNA. This binding is facilitated by a molecule called elongation factor TU. Elongation factor, EF represents elongation factor, TU helps binding of a charged aminoacyl-tRNA molecule to the A site. Next, a peptide bond formation will occur between amino acid at A site and amino acid at the end P site. This blue represents actually a polypeptide chain. Here numerous amino acids are joined together. The end amino acid in this polypeptide chain will form a bond with the amino acid at the A site, and this is facilitated by peptidyl transferase enzyme, and for this the energy is provided by GTP molecule. Next, now after formation of peptide bond, there is a shifting of position of this growing polypeptide chain from P site to A site. You can see in this picture, here P site tRNA is vacant while A site tRNA contains a growing polypeptide chain. This P site tRNA is now termed as uncharged tRNA. It is not having any amino acid or any polypeptide. Now it can leave the ribosomes and it can attach us with another amino acid for the next round of elongation phase. In the A site, now we have the growing polypeptide chain. Next is the translocation. In this translocation, the ribosome will shift its position. When ribosome move along the mRNA molecule, naturally the position of these tRNAs will shift. Now the tRNA at the A site comes to the P site and tRNA at the P site comes to the E site. This E site tRNA will leave the ribosome and attaches with another amino acid. During this step, elongation factor TU and GDP will also dissociates from this complex, and at this condition, this elongation factor is inactive. Only with the presence of GTP molecule, elongation factor TU can facilitate the movement of or attachment of an aminoacyl-tRNA to the A site. But in this condition, it is inactive. So there is another component called elongation factor G, EFG. It can convert the inactive elongation factor TU to active form. It will attach a GTP molecule with the EFTU and makes it active so that it can carries out the next round of elongation phase. This process will continue until the termination codon is reached. Next is the last phase, that is the termination. In termination, it is signaled by 3 stop codons UAA, UAG and UGA, occur amber and opal codons. These codons mark the onset of termination of translation. When these codons are reached, there is no addition of amino acids. There is no tRNA specifying amino acids for these codons. There are some other factors that act during this step. They are called release factors. RF1, RF2 and RF3 are the 3 release factors which are involved during termination of prokaryotic translation. RF1 and RF2 are specifically involved in stopping the process, and RF3 is involved in release of the 30S and 50S ribosomal assembly. Another factor is peptidyl transferase. This peptidyl transferase enzyme is required for release of the polypeptide chain from the ribosomal complex. This is the picture showing the termination process. Here, when stop codons reach, here no tRNA will be attached. Here release factors will attach at the A site. This will signal release of the polypeptide chain from the P site, and this release is facilitated by the addition of water molecule to the end of polypeptide by the enzyme peptidyl transferase. When release factors are attached to the A site, peptidyl transferase enzyme will add water molecules to the end of growing polypeptide chain, and as a result they get released from the peptidyl tRNA. Next, IF3 will bind, and this IF3 GTP assembly will dissolve 30S, 50S ribosomal assembly. Now they also get separated and they can now involved in next round of translation process. So we are coming to the end of prokaryotic translation. Here is the summary. First is the formation of initiation complex. Here mRNA 30S ribosomal subunit, 50S ribosomal subunit and fMet-tRNA form an assembly, and this is called a initiation complex. Following this, next elongation will continue. In elongation, a growing polypeptide is formed by the addition of amino acids, and next is the final step, termination. During this step, the newly synthesized polypeptides will be released from the ribosomal complex. Hope you all understand this. If you have any doubt, you can ask by posting comments. Thank you for watching.